Hello.
Hello, Mikris.
Hi, Ren. How are you?
I'm fine, thank you.
I hope you can see and hear me clearly, right?
Yes, but if you want, you can also check the share screen function.
Oh, yeah, sure, definitely. Let me open my presentation here and let me share the screen. In- Can you see the screen now?
Yes, yes, it's very clear.
And also, when I start the presentation, can you see this screen changing now?
Yes, yes. Yeah.
Perfect.
Yeah. Thank you very much.
No problem. So I will join back around just before 10 with the same link.
Yeah. And do you know Professor van der Oost? He will join from the same computer, or he is?
I think he is in his home and I'm at my home because of the COVID. We are not allowed to be in the lab unless it's, yeah, I think John is here. So,
Okay. Okay.
Hi.
hi, John.
Hello, John. How are you?
Can you see me and can you hear me?
Yes, we see you. You are very nice, very young.
Well, it's great to connect to you, and I'm looking forward to interact with the people in Romania shortly.
Thank you. So we have something like 170 registered participants covering from Azerbaijan, Romania, from different other parts. So yesterday it was
How many did you say?
170.
So how many did you say? 170. Okay, that's great.
Yes, registered. Yes, and they are covering a very large, very broad geography from Azerbaijan to Turkey to Greece to Romania and Ukraine. So it was a very significant interest on particularly your lecture. And yesterday we had, let's say, the introduction to this course from the perspective of microbiology in the Black Sea waters, the aspects related to the, let's say, problems of detecting different types of possible epidemic diseases in water, and the way in which this activity is organized. And this was the, let's say, what is the danger of swimming in the water. And now today we would like with your presentation to show how it is possible to put these microbes to works and the algae and things like that. Of course, it is a long way.
It is a long way, and I think well, of course, I mean, the CRISPR technology offers a lot of possibilities, but it's not, or that's my opinion at least. We are not going to change the entire world. So I mean, I don't know if we can really make sure that we can reduce the toxicity of certain algae or cyanobacteria or whatever. But for sure, in biotechnology and in biomedicine, so to say, there are huge opportunities. So I think that's really nice.
Yeah, from the other perspective, I am a member of the expert group on nanomaterials at the European Chemical Agency, and we are looking to the aspects of risks and vulnerabilities due to these nanoparticles that are manufactured industrially and are now already present in many products. So the problem that we are facing from chemical point of view, for instance, is that every new year you have 4,000 new type of synthesized substances that are generated out of research activities and so on. So how can you organize the whole, let's say, testing, security, in order to see if those new types of substances do harm the environment, harm the human body, and so on? So this was a huge problem, and it is very clear how difficult is to manage this. And now coming with these new technologies like CRISPR to effectively generate new type of, let's say, biological structures and organisms due to this new type of approaches, this is opening a new other Pandora box of managing and taking, let's say, to some sense, in a livable way, all these problems. But,
now, of course, I mean, there are a lot of ethical discussions ongoing, and also at the EU level. So the European Union, for instance, when it comes to using CRISPR for engineering plants, right? And there, well, we expect within a couple of weeks, we expect again a kind of a release, an opinion from the European Union, and hopefully they say that they will reopen the discussion, because a couple of years ago the European Court of Justice decided that CRISPR is the same as GMO, and that is very bad. And I think also, if you think about the future and about the countries where people don't have enough food, I think we really, as scientists, should take our responsibility to speak out that this CRISPR technology is much more accurate, much more safe in that respect, if you compare it to the currently used classical engineering or classical mutagenesis approaches, right? But before we go into a lot of details, maybe I should test my my slides before the meeting starts, right?
Yeah.
So let's try to see. So how can I upload?
So there is a share screen.
Oh yeah. Okay,
You just have to open.
So does this work for you?
Yes, very nice, very nice.
And should I? Okay, I try to. Okay, I guess this is okay. Okay, so shall I do it like this, or?
Yeah, it's working very well.
Well, is it? No.
But I think you have to press the
yeah. Okay.
Now it's fine.
Yeah. So now it works, and now also the animations seem to work. Okay.
Yes.
Now that's okay. So this is okay. So I go back to the first slide. So I will give a presentation between 30 and 45 minutes or so. Then maybe that's up to you. We could have a short break or directly continue with the talk of Miris. So what do you suggest?
Just 1 second, because I'm also on phone with Professor Kifiriuk. She's a professor from the University of Bucharest. So, doamna profesor, puteți să intrați acum în deschidere? Da? E de la 11, dar acum e 11. Da. Ok. Deci vă așteptăm. Bine. Da. Okay, so thank you very much. So you said that you're gonna make the first part of the presentation for about 30-35 minutes.
Yeah.
And then we shall have a discussion on this part. And then Mihris is going to continue with the part of Algaea and the biotechnology part.
Yes.
Sounds good.
Yeah.
Perfect.
Because I mean, of course, we can also give break, but the chance to have you with us is very unique.
Yeah. Okay. No, that's
and
fine.
we want to take the advantage of
sure. Okay.
discussing with you.
Okay.
And putting questions and so on. So yeah, you mentioned very well that there is also a responsibility on behalf of the scientists to show the evidence and to offer, let's say, all aspects in such a way that the decision makers to take it. But they have to take the decision knowing all the aspects. And
that's exactly right, and that's a very important point, and that's why I also try very hard to reach out to the politicians and to make them aware of what we are talking about. And I think that every political party that has a stand in this discussion should have specialists on board that know what it is about, right? Only then you can make a good decision. And when they know what it is about, they also must agree that it's ridiculous that they now say that this is much more risky compared to the ongoing technologies. So.
Da, s-a trimis. Haideți că o rog încă o dată pe Andreea să vă trimită încă o dată.
Should I leave the screen as it is now, or should I?
Please, please don't leave it as it is.
Okay, that's fine.
Yeah.
I just go and get a glass of water before I get started.
Please.
So I'll be back in a minute.
Yes, thank you. So once again, hello, Professor van der Oost. I would like to welcome also Professor Carmen Chifiriuc. Professor Carmen Chifiriuc is coordinating the activities on microbiology at the University of Bucharest. They have also a hub for innovation, the InnoCube, and they are developing many other activities in this area of microbiology. And of course, from that perspective, it is a chance that we have also the presence of Professor Chifiriuc. So if you would like to say a few words at the beginning.
Yes, of course. Can you hear me?
Yes, we can hear you very well.
Okay. Dear colleagues, dear students, dear friends, first of all, I would like to thank Professor Mahmoud for inviting me to participate in this session and congratulate him and his colleagues for this great initiative. This initiative, I think that responds to an acute need and at the same time a great expectancy that our society has from us, respectively educating our students for being able to respond to the local needs, for contributing to regional development strategies, for addressing major societal challenges of the 21st century by using research-based results, which are obtained by breakthrough concepts and methodologies and translating them to innovation. And I'd like to thank Professor Mahmoud that he's helping us at the University of Bucharest to bridge research and innovation through the MedGreen cluster, in which our university is a member too. And speaking of breakthrough science, it is an honor for me to attend this talk on CRISPR-Cas technology held by Professor John van der Oost, the pioneer of one of the breakthrough concepts in the biological research represented by the CRISPR-Cas revolution, allowing the development of genome editing applications in many fields, from basic research to different practical applications in different fields of our activity, or from ecology to medicine. I'd like also to share with you that University of Bucharest, besides being a member of the Black Sea Universities Network, it is a member of the CIVIS European University. And within CIVIS, together with our other 7 universities from Europe, we have identified some main challenges of global challenges for the 21st century. And I think that this course is related to at least one of them. So these major challenges are represented by health, cities, territories, and mobilities, the digital and technological transformation, and society, culture, heritage, and climate, environment, energy. And I think that this course is in the frame of these major global challenges identified by this European Universities Consortium. At the same time, I'd like to say a few more things about the hub developed at the University of Bucharest in the frame of the Research Institute of the University of Bucharest. This Research Institute of University of Bucharest has been established in 2014 to stimulate and promote excellence in research activity by assuring a dynamic environment for both research projects and careers, and encouraging international collaborations. We are offering a common platform for organizing international scientific events like courses, short-term courses, workshops, but also conferences, national and international conferences. We are encouraging interdisciplinary research area, and also we are offering fellowships for postdocs, but also fellowships for invited senior researchers and professors that would like to come to our university and share their research results and transfer good practices and know-how to our university. So all students and researchers present here are invited to start to look at the website of our university, of Research Institute of the University of Bucharest. And now related to our focused interest on this presentation, in our lab from the Faculty of Biology, we are trying to introduce the CRISPR-Cas methodology for fighting antibiotic resistance, because this, the study of antibiotic resistance in clinical and environmental sectors are one of our major fields of research. We are studying antibiotic resistance by phenotypic and genotypic methods, but also we are evaluating and developing new strategies to fight antibiotic resistance. And we have a PhD student who started his PhD 2 years ago. And his focus in the PhD thesis is to use the CRISPR-Cas technology for removing carbapenemase resistance genes in Acinetobacter baumannii. That is one of the most fearful resistant opportunistic gram-negative bacteria. I'm eager to hear Professor John van der Oost. He has a huge international recognition great scientific personality, proved also by the over 20,000 citations that he has for his works in the Web of Science. So, Professor van der Oost, you have the floor.
Well, thank you very much for this very kind introduction. It's a great pleasure to reach out to Romania and other countries in the area, and it's a great pleasure always to talk about CRISPR, about the fundamental features, but also about the possibilities to use these fundamental insights. So what we will do today, so me together with one of our great talented PhD students in the group, Miris, we will give basically, or we will split the presentation into two. So I will start with a kind of historical overview, then move to the mechanism, and at the end we'll say a few words about applications. And then, of course, there will be ample time for discussions, for questions, and then Miris will describe the research that he has been doing over the last 4 years in our group on microalgae. So using the CRISPR technology for engineering microalgae, and basically, as you will see, he invested a lot of time to kind of build a toolbox for at least some of the microalgae.
So, okay, Professor van der Oost, before starting, allow me just to remind to our participants. So there are almost 80 participants that are following now. There is a button for question and answers. So please use this button and post your questions or any kind of comments that you will consider. And then we shall have 2 sessions of discussion. The first one will be after the lecture of Professor van der Oost, and will be moderated, both will be moderated by Professor Chichiriuc. So we shall have a first part of the discussion on the conceptual aspects of CRISPR-Cas technology. And in the last part, we shall discuss more detailed about what can be done, and even for some project proposals and cooperation opportunities. So once again, thank you, Professor van der Oost. You have the floor.
Okay, thank you very much. So let's. So this is basically what I already said. So history, biology, applications. So let's start with the history. And before starting with a description of CRISPR-Cas, we should go back to the basics because, as you may know, CRISPR-Cas is a defense system by bacteria and archaea to defend themselves against viruses. So what happens? So viruses not only attack humans, as we now all are very much aware of, but also plants and other eukaryotes, but also prokaryotes. So also bacteria suffer from specific phages, as we call them. And here you see what happens upon recognition of a specific host. Then the genetic material of the virus or the phage is injected. In this case, it's double-stranded DNA, as you can see here. That DNA is then replicated to generate new DNA, but it's also being transcribed and translated to protein, and those proteins make up the capsids, and altogether. So the capsids, they package the newly formed DNA, and at the end that gives rise to complete new viruses. So such a cycle can last between 30 and 60 minutes or so, depending on the conditions. And then new particles are being released, and then we are talking about 20 to 100 new viral particles on average. And then they get out of the host cell by also producing some enzymes to break the cell wall, and that often leads to the death of the host. And then the new generation of viruses, they start looking for a new host, and then the cycle continues until these bacteria, they meet resistant, or these viruses meet resistant bacteria. And already for a very long time, several mechanisms by bacteria to keep out the viruses are known. And basically, what the list that you see here, they are different mechanisms that operate at all levels of the viral proliferation cycle. So starting with the attachment and the inhibition of the DNA injection, but also, and this is probably the best known system, so that's the restriction modification system, so that can recognize alien DNA. So it can discriminate the self DNA. The chromosomal DNA is not depicted in this cartoon, but it can discriminate at that level, so it can recognize the invader DNA and then degrade it. And when all these systems do not work, then there is kind of a last rescue system. Well, rescue for the population, that is, because it's a suicide system. So then the cell sacrifices itself to avoid the release of viral particles by committing suicide. So it's a cell death system. And then there was the discovery some 15 years ago of this potential new antivirus system, CRISPR-Cas. So how did that happen? Well, basically, the first time that part of the CRISPR system was found was already back in 1987 by a group in Japan, and it was a student, Yoshi Oshino, who found this sequence in the genome of E. coli. What he was doing, so here you see again a kind of a cartoon of the chromosome of E. coli. He was sequencing this region. He found the gene that he was interested in, but he also looked at the region downstream, and then he ended up in this sequence. And he immediately noticed these repetitive repetitive character of this sequence. So highlighted here, you see the repeats of the CRISPR arrays. So later on, the name CRISPR was coined by, in fact, by a group in the Netherlands together with a group in Spain. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. So here you see the repeats highlighted, and in between those repeats are the spacers of the CRISPR, and they are indicated here with different colors, also here, to highlight that those sequences are completely different. And also the same group in the Netherlands, Ruud Janssen and his colleagues, they found that adjacent to these CRISPR arrays, usually there are clusters of genes, and he called them the CRISPR-associated genes, or the Cas genes, coding for the Cas proteins. And then a very important finding, initially by a group in Alicante, by Francisco Mojica, but later on also by two groups in France. They found that, in fact, maybe the secret or the answer of what the functionality of this CRISPR-Cas system was, that can be found probably by looking at the spacers, because these three groups independently found that at least some of these CRISPR spacers were copies of fragments of viruses and of plasmids. So that led to the hypothesis that maybe we were dealing with a novel defense system in bacteria and archaea, and they started to speculate. These groups started to speculate about a functional mechanism that might resemble the eukaryotic RNA interference system. So where small RNAs are being used to attack RNA sequences, for instance, of viruses. So also, at least partially, also as a defense system. But I will come back to that. Also, so from the 1990s onwards, many bacterial and archaeal genome sequences were completely established, and it was found that in about half of these prokaryotes, CRISPR-Cas systems were present. It was also found that there was a lot of diversity. So this is the most recent classification, and so we now recognize two different classes, and I will come back to that in detail in a couple of minutes. So two classes. One and each of these classes, they contain three types and many subtypes. So new subtypes are still being found in genome sequences of bacteria and archaea. What I will discuss today is the DNA targeting CRISPR-Cas systems. So they are highlighted here. But apart from that, there are also at least two. So type 3 and type 6, they are targeting RNA, but I will not talk about that today. But of course, if you have questions about that, I would be happy to try to answer them. Okay, so let's after this introduction, let's now move to the biology. So how does the system work, and how did we figure that out? So here again, you see a Cas operon, so a CRISPR-associated cluster of genes next to the CRISPR array. So this is the CRISPR locus. And again, in black, you see the repeats, and in the different colors, you see the different spacers. So upon an attack of a virus, in this case, well, most of the infected cells will die after the proliferation of the viral particles. However, we also know, and that's for instance also known these days, if people analyze corona viruses like COVID-19, that in a subfraction of the viral population, mutants occur. Well, this also happens in the case of bacterial viruses. So there are always, because there is a relatively high mutation frequency upon the replication of the chromosomal DNA. So it may happen that a bacterium is infected by a mutant that at some level cannot efficiently replicate itself. Well, when that is the case, then the CRISPR system can act, because then it can, as I will describe in a three-step mechanism, it can try to gain immunity against this particular virus. And the first step is called the acquisition of new information. So acquisition of new CRISPR spacers. So what happens is that some of the Cas proteins, they select for a fragment of the viral DNA, and they process it into a certain length. And in the case of the E. coli system, for instance, it's between 30 and 32 base pairs. So to give you an idea about the size, those fragments are then going to be inserted in the CRISPR array at one side of the CRISPR array, and that so that works by opening up the system at one site, to duplicate the repeat that is located there, and then to insert the new part of the new part of information that is important for the immune system. So basically, now this is called adaptation of the CRISPR array, and basically what happens is that a piece of information is added to the memory of the immune system. So this is really, or this was the first example of an adaptive prokaryotic immune system. Then the second step is that the information is being expressed. So a long precursor CRISPR RNA is being generated, and then often together with the proteins that are encoded by this cluster of Cas genes, they assist at different levels of the whole mechanism. So in the case of the E. coli system, for instance, it's a particular Cas protein that is responsible for the processing of this precursor. So then small RNAs are being generated. They are making a complex with yet another Cas protein, and that allows for recognition. So when there is an infection by a phage of which information is already stored in the CRISPR array, then specific recognition can occur, and then together with yet another Cas protein, then neutralization of this invasion can happen, because then after recognition and recruitment of this particular nuclease, the incoming DNA, so the alien viral DNA, will be destroyed, as we will see in a minute. But there are some potential problems, like with every immune system. There are problems of autoimmunity, and that can happen because the sequence that is being recognized by this CRISPR system, that exact same sequence is of course also present on the genome of the host itself, right? And so this CRISPR spacer is derived from a protospacer. That's the name for this particular sequence that is being selected. So the protospacer ends up as a spacer in the CRISPR array. But of course, this exact same sequence that will be attacked at the end is also present on its own chromosome. So how is that self DNA being discriminated from the non-self DNA? Well, that is by making use of the flanking sequences, right? Because the flanking sequences are different. So in the case of the CRISPR array, it's the repeat sequence, and in the case of the virus, it is the native flanking sequence of this particular protospacer or this particular target sequence. Okay, and that is indeed what the trick is of the system. So let's now zoom in, and that discrimination occurs already at the level of the adaptation. So only certain sequences are being recruited that can also act as an efficient target sequence. But let's now focus at this part of the graph. So let's zoom in. And then you will see, so here you see that the dashed blue line is kind of the Cas complex that binds in this reddish color the mature CRISPR RNA guide. So here you see leftovers. So the flanking sequences are the leftovers of the repeat. But the cleavage or the processing of the precursor CRISPR RNA occurred at this particular site, and in the middle is the important sequence. So that's the spacer part of the of the guide RNA, and that is responsible for base pairing, so opening up of the double helix. And then this is the important sequence for the self non-self discrimination. So that is what is referred to as a motif that is adjacent to the protospacer sequence. So it's called PAM, for protospacer adjacent motif. Okay, only when this motif is present, as it is in the virus, but not in the CRISPR array, because then you have the repeat sequence. So that will not contain a PAM sequence, but if that PAM sequence is being recognized, then the system will continue and try to degrade the DNA, so to generate damage on the DNA. So that only happens at the level of the viral DNA, but not at the level of the CRISPR array as part of the chromosome of the bacterial host. Okay, so so how does this look in E. coli? There is no particular reason for showing this cartoon except for the fact that I think it's very beautiful. And this is an update of a version that was already made 20 years ago. And the update, so they now included also, so this is part of the E. coli cell. Of course, it's an artist impression. Here, for instance, you see a flagellum integrated in the two membranes of the E. coli cells. And here you see, in fact, a CRISPR complex, as is depicted over here. So that's the what is now called, or that's the name we gave it, the cascade complex. And here you see a bigger picture of that cascade complex. So cascade stands for CRISPR-associated complex for antiviral defense. And so to end this part, I show you this movie. So here you see the cascade complex of E. coli with 11 different subunits. There are a couple of similar subunits there. So altogether, it's 5 different genes coding for these the subunits of the cascade complex. And in the middle, you see here the CRISPR RNA. So now it's looking for a match at the DNA level. And of course, you can imagine that the DNA is large, so it will take a while. But here, the PAM is found, and also a matching sequence between the guide and the target sequence. So the viral protospacer, that leads to a conformational change of cascade, and that allows for recruitment of this Cas enzyme that is going to degrade the viral DNA. So this enzyme is kind of a combined. So the green part is a helicase and the yellow part is a nuclease. And as you can appreciate, that leads to major damage in this case of the viral DNA. And that is what we figured out by studying E. coli. And I can talk for this for half an hour, but I don't have the time for that. But if you want to know, I have a couple of slides at the end of the presentation, which I will not show. But if you want to know the details, I can show them. So what we did is we used E. coli BL21, which does not have a CRISPR system of its own. We transplanted the genes one by one of E. coli K12, and we made a designed CRISPR against a particular virus. So we just randomly took fragments from four genes, and that's how we made a designed CRISPR. And this was then the combination at the end that worked. So 5 genes coding for the subunits of the cascade complex, and 1 additional gene of this helicase nuclease that I showed at the end of the movie. So that's called Cas3. So what did we learn then? Well, basically, and that was published in 2008, we revealed the molecular basis of the CRISPR-Cas system. We found that a maturation occurs of this precursor CRISPR RNA to in this case 4 mature, small CRISPR RNA guides, and that those guides were, in our case, bound to the cascade complex. But as we will see later, they can also bind to a big protein called Cas9 or Cas12. So that will form an effector complex, and then these systems use their RNA guide to specifically detect a sequence at the DNA level and then cause interference. So meaning that they disrupt that particular DNA sequence, usually by cutting it. What we also showed in this particular experiment, that we could transplant functionally the Cas genes and the CRISPR to other organisms. In this case, to another E. coli strain, but still we could functionally transplant the system to another organism. And last but not least, by using this design CRISPR, we could show that we could do multiplexing, meaning that we could target several loci at the same time of our target DNA. And that is basically not only providing the basis for CRISPR-Cas, the fundamental mechanism, but basically this also provided some first insights in how we could use this system for genome editing. And that is then bridging to the last part of my talk, and I will briefly describe the application. So genome editing, for instance, in biotechnology, but also, as you may be aware of, this technology is now also being used for trying to cure genetic diseases in human patients. Okay, so far we talked about this class 1 system, which consists of this cascade type complex. And that's true for all the class 1 types. So we only talked about one of them, and it was the E. coli type. But now we will move to the class 2 systems, because that is the well-known Cas9 type that is part of this class 2 system. And there you see that basically the functionality of these six genes is captured in a single big multi-domain protein, in this case Cas9. But as we will see, this is also true for another type, and that's called Cas12. And here, to compare the two systems, you see the class 1 cascade and the Cas9 system. Here you see the guide RNA together with a single-stranded target and all the different subunits of the cascade system. And here, in the case of the Cas9 system, it's one polypeptide with a slightly more complex guide structure, but still here you also see base pairing with a single-stranded DNA molecule. So here you see again the Cas9 system, and now I compare it also to Cas12, which is also a class 2 system. And so in red you see the guide molecule, as said, that is a bit more complex as the case in the cascade system, and also more complex than is the case in the Cas12 system. So this more resembles the guide in cascade. So part of it is responsible for base pairing with the DNA, and then this part is a leftover of the repeat sequence, and that functions. So you see a hairpin structure of the RNA, and it functions as an anchor to bind it stably to the protein. Well, in Cas9, as said, that consists of a CRISPR RNA together with a so-called tracer RNA, but a synthetic loop has been proposed or has been made by the groups of Jennifer Doudna and Emmanuel Charpentier to make it a single guide RNA as well. The big difference with the cascade system is that that you get a very clean cleavage of the double-stranded DNA by, in the case of Cas9, by two different nuclease domains. So the yellow and the green one responsible for cleavage of the double-stranded DNA target. In the case of Cas12, there is just a single nuclease domain which is responsible for cleavage of the two DNA strands. This is called, or the analogy with the Swiss Army knife is often made because of the all the different functionalities within a single polypeptide of Cas9 or a single polypeptide of Cas12. If we now again compare that to the cascade system with the Cas3 helicase nuclease, which causes a lot of damage, then it's clear that these type 2 systems are very useful for genome editing, whereas the type 1 cascade system is very useful to fight viruses and to neutralize and kill viruses at the DNA level, but it's not the first choice for precise genome editing. The good news, however, is that in the meantime we also made a version of the cascade system where we excluded the kind of chainsaw like Cas3 nuclease, but we replaced it by domains from a restriction enzyme, the FOC1 enzyme. So by doing that, we could obtain also in human cells. Now we could obtain very accurate and relatively efficient targeting of human target genes. So also that can now be used, but I mean, for obvious reasons, for practical reasons, these Cas9 and Cas12 proteins are the for most people are the first choice if they want to start genome editing. So if you want to use it for genome editing, of course, the first thing is that you select a certain site at the chromosome of the target organism where you want to introduce a change. So then you design a guide based on that sequence, and then you generate a double-strand break either by Cas9 or by Cas12, and that's basically where the CRISPR or the involvement of CRISPR stops, because then it's the repair systems of the cell that you're working with. It could be a bacterial cell or a plant cell or an algae cell or a human cell. Then the repair system of that cell is taking over, and it's trying to repair the break in the DNA, and it can do so by using a non-homologous repair system or by using a homologous repair system. Well, the non-homologous repair systems can be very useful if you want to inactivate a certain gene, or if you want, for instance, to reveal the function of a particular gene. But also for some applications, it could be useful to make a gene knockout. And then this is the system to go for. But if you want to do very accurate repair of maybe a single base pair, then you should go for the homologous repair system. Often people use, or they do not only introduce the nuclease with its guide, but also a repair fragment if you want to get this precision engineering. And that can be double-stranded or single-stranded. Both have been reported to work. And then you get really very accurate engineering. As said, that can be at the base pair level. It can also be a way to introduce a gene in a certain chromosome at a desired place. Okay, that brings me to the end of this part of the story. Um, uh, well, the good news is that Miris will take over after the first discussion. But let's first wrap up by providing some conclusions. So what I want you to remember for the rest of your life is that CRISPR-Cas is an adaptive immune system derived from prokaryotes. So it's just like restriction enzymes. Also, this CRISPR system is only present in bacteria and archaea. Now, the systems that we now know of, they all use RNA guides, so CRISPR-derived RNA guides. And what I talked about today is the systems that target DNA, but as said, there are also some systems that target RNA specifically. The diversity is huge. So 2 classes, 6 types. Well, I think right now at least 30 subtypes. So there is still a lot to be discovered at that level. The 3 systems that I focused on today, so Cas9 and Cas12, also to some extent the cascade, when it's fused to this restriction enzyme domain, they can be used for genome editing. And I think we all agree that because of the flexibility of the guide design, and so hence the flexibility of what you want to target, this really has led to a revolution, because several systems were out there before CRISPR to allow for genome editing, but this the flexibility in the guide design, that really has allowed for a revolution. And that is not only true at the level of biotechnology. So many examples can now be found in the literature on bacteria, yeast, plants, and we will hear much more about algae by Miris soon. So not only biotechnology, but also biomedicine, so clinical trials, and that's really very exciting, I think. Clinical trials are currently ongoing to cure some human blood diseases. Ex vivo, the editing is being done. So they take the blood cells or stem cells for those blood cells with a genetic mistake there, and they repair them so ex vivo, so outside the body, and then they do the repair with CRISPR. They check if everything went okay. So if the desired change has been made, and if there are no off-target problems, then they put the stem cells back in the bone marrow of the patients, and that is really ongoing. I heard that there are some first cases where that happened successfully, so that the patients were cured of that disease. And I think this is only the beginning. We will hear many more of those cases, hopefully in the near future. Then, if you want to see a nice movie about the CRISPR discoveries, you should have a look at this. It takes you basically what I now talked about for half an hour. They describe that very nicely in 3 minutes. So I will make this presentation available to you, so you don't have to make notes. Maybe I should have told you before, also this link you will get then, and then you will see that also these two ladies are being mentioned in that movie. So that's Jennifer Doudna and Emmanuelle Charpentier. They, as you probably know, they received the Chemistry Nobel Prize last year for their work on Cas9, and because of the huge impact of Cas9. These are the people that actually did the work. So this long list is the people in Wageningen, and some of the pioneers in the group you see over here. So these guys were involved in the discoveries that I described on the cascade system. but these are the people, the talents that, or part of the talents that are currently working in the group on CRISPR. And what you see over here is many fruitful collaborations we had over the years with many groups. So few in the Netherlands, but also several groups outside the Netherlands. And with that, I come to the end of the presentation. So if you have questions or remarks, I would be happy to hear that, and I will do my very best to provide some answers. Thank you.
So thank you very much. Really very special presentation, and all that put in a nutshell. So for the understanding of a large and broader audience. So I would kindly invite Professor Kifiriuk, if you would like to take the lead for this first panel discussion.
Yes, thank you, Professor Van der Oost, for your presentation. Very informative presentation, very synthetic presentation on this major discovery of biological research. Now the session is open for discussions. So I'm looking in chat and question and answers, but I see no question here. So don't be shy if you have questions for the speaker.
Just for starting, there is a question.
Okay. So how is CRISPR-Cas9 delivered in human cells, and how long does it take for it to work?
So can you repeat the question?
How is CRISPR-Cas9 delivered in human cells?
Okay.
And how long does it take for it to work?
Okay, well, that's of course a very important question. So the question is, how is Cas9 introduced in human cells, and how long does it take to act? Well, the delivery of these systems, that indeed is a major bottleneck. And that is something you have to figure out by trial and error. What people use mostly these days, and we don't do that, or we do some work on human cells now, but I mean, we only started with that a couple of years ago, but people use the electroporation technology, I think most of the times. So there you produce the protein, for instance in E. coli, and you make the complex with the guide, and then you use electroporation of the human cells to get the protein in. There are also some other techniques to get the protein RNA complex inside the cell, and so there are several reviews out there. If you want, I can come up with a couple of reviews that really address that. The alternative is that you introduce it as a plasmid. So again, by electroporation, you can introduce it, but there are also other techniques. And then of course the expression of the protein has to occur in the human cell, and for that they then often optimize the codon usage of that particular Cas9 gene to allow for efficient production of the protein at the end in the human cell. Of course, also you should not forget to add a nuclear localization signal, because that is or that increases at least the efficiency of the import into the nucleus where you want it to act. And then the question, how long does it take to see an effect, or to have an edit? Well, people did a calculation once. If you have a single Cas9 protein molecule inside a human cell, then it will take I think 2 days for it to find the right target. But of course, if you have 100 molecules, then you significantly reduce that time. And so then you can easily get an edit after 1 day or after 2 days. Of course, you usually want to have an edit at both copies of the chromosome, where but so that is so 1 or 2 days. Then with most technologies it should work. But I should also say that it all depends on what kind of human cells you use. So I mean, different cells have different structures and different features. So it's all a matter of trial and error. But by now there are many examples in the literature, so that could give you some ideas of how to tackle a certain engineering approach in a particular cell type. More questions?
There is one question from Adrian Ionaș. Which is the more efficient method for gene editing in organisms, such as the meganogaster? Should it be more efficient when working with embryos or full-grown adults?
I'm not sure that I got the question. So this was about Drosophila?
So in principle, so is for gene editing in organisms.
In Drosophila melanogaster, yes.
Yeah. So what is the question? If it should be done in embryos or in adult organisms?
In adults, yes. Which is the more efficient method?
Yeah. Well, I mean, it's obvious. If you want to edit all the cells of a particular organism, of course the best way would be to do that at a single cell level. So after the egg has been fertilized, that should be if you then get the edit you want, you can assume that it will be in all the cells of the organism, and that's true for Drosophila. But of course, it's also true for humans. So if you really want to tackle serious diseases, or if you want to make sure that future generations do not suffer from a certain genetic disease, you could consider if that's allowed, because that's another thing. But I mean, the best thing would be then to do that at an embryo level. But then, of course, there are well, first of all, ethical issues, but there are also safety issues. So about the ethical issues, well, we can talk about that. But about the safety issues, of course, it has to be 100% sure that you get the edit that you want, and that should be checked, especially if it comes to human embryos. And then when you get the exact edit that you desire, and you do not get any undesired off-target effects, then I don't see any problems, especially so when it comes to ethics, especially if you realize that already embryo selection is ongoing as we speak, right? At many places all over the world, in hospitals, they do the in vitro fertilization of human egg cells, and they when parents know they are carrier of a certain disease, they can go for that option, and then they can have it. So they then have a series of egg cells that are being fertilized, and then the specialist can take, I think, from the 8-cell stage, they can take one cell and check it, and then if that has the desired genotype, they can select that to implant it into the mother, and then you have, when all goes well, you have a baby without the genetic problem, right? So, but again, that's an ethical discussion. That's what at a government level has to be decided. Do we want that, and do we allow that? And is the technology safe enough? That's a very important issue. But then that's how we can deal with that. And of course, I mean, coming back to the question, if you want to do this in Drosophila, and if you have the possibility to do it at the embryo level, I would certainly do that.
So thank you.
Yes, I think we have more questions.
Okay.
Okay. How effective could be CRISPR-Cas9 turn out to be on eliminating COVID-19?
Yeah, that's a good question. Well, what I know for sure, and also in our group, we are working on diagnostics of using CRISPR systems to detect COVID, and that works very nicely. So systems have been described already in the literature based on Cas9 and Cas12. But in our group, we recently also established a system using a cascade system that targets RNA. So that's the type 3 that I mentioned earlier on in my presentation, and that also works fine. But of course, if you want to use the CRISPR system to get immunity in humans against the COVID, that is going to be very difficult in my view, because then basically you should have the CRISPR system in all your cells, right? And then in principle it should be able to work, but then we really have to design the human genome again to make that work. So I think that's not an option, so I think we better use the technology as we have it now to produce many different vaccines and try to vaccinate the whole population as soon as possible. And I think that's the way to fight these nasty disease. But also, I mean, let's face it. I mean, a year ago the genome of COVID was released. And look at what happens now. There are at least 4, 5, 6 vaccines that are being used all over the world now, and many more are coming. So that indicates that really we can react very fast to something like this, and I think that's really very impressive, and that's again highlighting the importance of fundamental research, just like we described today with CRISPR-Cas, but also at many different levels. And I think it's beautiful. Of course, it's a nasty disease, but it's very good to see how fast now the industry can react to come up with these ways to fight the pandemic.
Don't you think it is a little bit too far reaction? Wouldn't it be better to have a period of testing all these vaccines and have, let's say, a broader understanding of possible impact of these kind of vaccines?
Of course, I mean, you have to do safety analysis, but I think all the vaccines that are out there now, they have been tested, and of course there are some problems, but I mean that's true for every medicine that is on the market. Even aspirin has off-target effects, right? So of course there are people that have some risk, but this morning I read in the newspaper that also the Zeneca vaccine, for which there are some reports that people have some thrombose like issues. That also there, it's 1 out of 150,000 that at least based on the currently available numbers, that have this problem. Of course, it's nasty, it's terrible for these people that have the problem, but all the others, they build up the immunity of the population. I think with these numbers, if that turns out to be true, then I think we should go on as fast as we can with vaccinating the entire population.
Thank you. You mentioned that you already are developing in your group some research on detection of the COVID virus. So a problem that we have been facing, we had also some work with a group from the University of Bucharest. It was the possibility to evaluate the activity of the virus. So the potential, the infectious capacity of the virus. For instance, you have a virus that it stands on different substrates, on different surfaces. And sometimes there are some, let's say, estimations that the infectious capacity of the virus is just for a limited number of hours. So would it be possible by this kind of technique to evaluate the viral capacity? So you have virus, you can detect it, but if it is still possible to infect other molecules?
Yeah, that's a good question. I don't know enough about that to come up with a real answer, but so that it in theory it should be possible if you know what changes at what level. Is it at the protein level that maybe I guess it is. So I guess that there should be maybe some proteins that are responsible for finding a way to enter a human cell. So to find the receptor. So it's probably the spike proteins that don't have have a certain half-life, so to say. So and if you can indeed, then you should know more about the denaturation of those spike proteins, I guess, to have antibodies that can detect different stages of that denaturation process. Then it should be possible. But I mean, the system that we developed is basically detecting the RNA of COVID, and that of course can only be used for at best detecting mutants, for instance, and that's the beauty of the CRISPR detection systems. So focusing at nucleic acids, that we can find single nucleotide differences between mutants, and so that's I think very good, and it's very easy to adjust the system by making use of this guide flexibility, right? So it's very easy to change the specificity and to be able to detect any mutant that we like.
Thank you. So, Professor Kifiriuk?
Yes, thank you, Professor van der Oost. We have two more questions.
Okay.
One of them refers to the possibility of using CRISPR for manipulating chromosomes. For example, for deleting or making non-functional an extra chromosome responsible for one syndrome like Down syndrome or Patau syndrome.
Okay. Again, well, this is an interesting question. But of course, I mean, if you want to do that, in a patient that is going to be impossible, I would say, because then you have to edit all the cells of that patient. You could do it at an embryo level, in theory, but I mean it's what is it? Chromosome 21, I think, that is there in 3 copies, right? So you have to somehow then discriminate or attack only one of the 3 copies, and I think that may be difficult when they are exact same copies. If not, so if it's known that, and of course I guess I don't know how the 3rd copy is being made. So if there are differences between this particular chromosome and the 2 sister chromosomes, so the other copies, if the 3 copies of chromosome 21 have differences, and I guess they should have because of crossing over to generate them, then it should be possible. But only I think this only makes sense at the embryo level, and it's a major operation to do that. So I think it would be much better than to go for the embryo selection that I mentioned before, and to select embryos which do not have this problem. I think for CRISPR, this is a bridge too far, I would say.
And the final question, Professor Kitiriuk.
Yes, I'm sorry, I had some problems with my mic. The last question is when can we expect a widespread of using CRISPR in different treatments for combating multiple diseases and illnesses?
So the last part of the question, can you repeat that? So using CRISPR for
different treatments for multiple diseases and illnesses. When we expect
okay, yeah,
that?
well, I mean, so what I mentioned at the end of my presentation is that it's currently being used for blood cells, because of course that's relatively easy. You can get hold of these cells easily. You can so you should take stem cells out of the bone marrow, no, I wanted to say I have a slide on that, but I can, it's easier to explain it. So you can take those cells, you can do the edit, you can check it, you can cultivate them and bring them back into the patient. And probably before you bring it back, you have to get rid of all the remaining stem cells that have the mistake in the body, and then bring it back. That all sounds very scary to me, but it seems to be routine procedures. So that is the trick that works. So, and what they did so far is they tried to cure sickle cell disease, but also another disease that is related to a point mutation in the one of the hemoglobin genes. So that works, but then of course what you really want, or for many diseases this is not possible. For instance, a muscle disease, right? The muscles are all over the body, and then what you want is to try to edit as many cells in the body as possible, and for that you should use viral delivery systems like AAV, for instance. So the adenovirus-based systems, and those systems have a limited capacity because it's basically using viral capsids. They have a certain size, and the genome cargo cannot be more than 4.8 kilobase or so, something like that. So that's basically too small, or that capsid is too small for Cas9. But the good news is that for Cas12, many variants have recently been found, and they are smaller, and they also do the editing. So currently we are also working on some of those systems in collaboration with people that are studying Duchenne. So that's one of the nasty muscle diseases, and so we try to make that work. Other possibilities, and that again makes use of some blood cells, is to really fight cancer. So by really equipping those blood cells, or making them better suitable to detect and to fight cancer cells. That's also ongoing. That's promising. But having said that, I think we should not make it too big. It's not going to happen that we can cure every genetic disease. So we should be realistic, and I think we should say that it would already be great if we can use it for some diseases, but we have to be realistic and not make it too big, I'm afraid.
So it is not possible to build a Superman yet.
Everything is possible. Also building a Superman would be possible, but I think talking about ethics, I think that's unethical. And if you, my advice to people that want to have the perfect baby, then I think they should not use CRISPR, but they should just select their partner to make that happen.
Thank you, Professor van der Horst. Related to this question and related to efficacy of CRISPR-Cas and translation to clinics, what is your opinion about translating into the microbiology field? For example, for fighting, as I told you before, antibiotic resistance or some kind of dysbiosis or some kind of very, very virulent bacterial pathogens. Do you think that it could be more easier than working with mammalian cells?
Yeah, well, I think I'm a microbiologist, so I, for me at least, working with bacteria is easier than with mammalian cells. And to come back to your question, can we use CRISPR to fight pathogens? I think there was already more than 5 years ago a paper where they tried to use CRISPR to fight some nasty strains of Staphylococcus aureus, and they did so basically by using phage therapy. So they equipped, or they got a couple of phages that could use Staphylococcus as a host, but they equipped that phage with Cas9 and a guide that specifically targeted the toxin genes, one gene or more genes, and that worked very well. So I think that could be a way to go. So I'm a big believer of phage therapy. I think that's a way better option than trying to develop antibiotics, and because I mean, because of the ongoing arms race, there will always be mutants that can find a way around it. But the beauty of phage therapy is that the phages kind of co-evolve with the bacteria. So there will always be phage variants that can deal with a new invention of a pathogenic bacterium. So I think I'm a big believer of the power of evolution, and in that respect also phage therapy. I really hope that will help us fighting pathogenic bacteria, and especially if you can further improve the specificity so that you do not kill all bacteria from a certain genus or certain species even, but only go for the strains that are really the bad guys.
Thank you, Professor. I'm totally agree with you. I see no more questions. So, Professor van der Oost, you have the floor for the second part of your presentation. Thank you so much.
Okay, well then it's the turn of Miris. So I can briefly introduce Miris. So Miris is not only a very nice guy. He's coming from India originally. He did his master thesis in Wageningen University, and we were the lucky ones because he selected microbiology to work there, and he did some excellent work on the Cas9 protein from a thermophile, from a thermophilic bacterium. And we are still working on that. And after finishing his master degree, which gave him, on the basis of his master's thesis, he obtained a prize of the best thesis in Wageningen of a master thesis. And then he started working on a project together with a group in Wageningen, led by Professor Maria Barbosa, and they are focusing on microalgae. And we were happy to be involved there, and we were even more happy that Miris decided to take the position, and he's now going to kind of in the next 30 minutes or so to give an overview of his efforts and the tools that he developed based on the CRISPR-Cas system. So Miris, take it away.
Thank you, John. Thank you for the introduction. And I would also like to thank Professor Mahmood for giving me this opportunity to present my almost more than 3 years of research work on developing genome editing tools. So I will share the screen now. I think, John, you have to stop sharing your screen, only then I can share my screen. Perfect. I hope my screen is visible now. Yeah. A very good afternoon. Good afternoon to all the audience from Romania. It's almost past 12 there, and it's just past 11 here. So let's go. So the PhD thesis is what I have been working on for almost a little bit more than 3.5 years, was to develop CRISPR-Cas based genome editing systems for a particular microalgae called as Nanochloropsis oceanica. Microalgae in general, they are phototrophic organisms. They can make use of sunlight and the atmospheric carbon dioxide to produce valuable compounds or metabolites, and also basically produce biomass. The valuable compounds that they produce include polyunsaturated fatty acids or omega-3 fatty acids, which are like high value compounds, and they are also capable of producing very low value compounds such as biofuels. But the present amount of biofuels that they produce, or the present amount of triglycerides, which is the precursor of biofuel production, what they produce is less, and the production cost cannot compete with the non-sustainable sources that are available out in the market. However, there are certain techniques like when you stress microalgae with nitrogen or any other kind of stress, they basically accumulate a lot of lipids or a lot of triglycerides, which can be used for producing biofuels or which can help us commercialize these low value compounds. But when you provide this kind of stress, what happens is that they grow very slow, or their biomass production comes very down, and the relative or the overall productivity of lipids are still low to produce large amount of lipids from them. So what we really want to do is to make use of microalgae, or to improve microalgae to make use of sunlight and the available CO2 much more efficiently, so that they perform or they produce large amount of lipids or triglycerides, and also they produce large amount of biomass. From from a perspective of a biotechnologist, there are two ways that we can try to develop microalgae to this end. So one is the classical or the random mutagenesis, where you introduce random mutations in microalgae, and then you develop selection techniques to select strains of your interest. The other one is to understand the metabolism of the species and perform targeted mutations. You look at the metabolism, introduce mutations at the regions you want, which is called as metabolic engineering. For both these approaches, we will require genome editing tools at some point. Especially for metabolic engineering, we need precise genome editing tools for introducing mutations or introducing certain pathways or genes. And even for the random mutagenesis, you will need tools when you isolate a particular mutant from your selection. You will need genome editing tools to further confirm that this is the real reason why you are having an improved trait. So this leads us to the CRISPR-Cas based genome editing in microalgae. So as John already mentioned in his talk, Cas9 and Cas12 with the type 2 CRISPR proteins are the most widely used Cas proteins for genome editing across all the species. And as we can see here, John did mention about the non-homologous end joining based mutation. So Cas9 and Cas12, one of the differences is that uh, the Cas9 introduces a blunt end double-stranded DNA break, and Cas12 introduces a staggered end. And all these double-stranded DNA breaks, let's talk only about microalgae now, can be fixed by non-homologous end joining, which can result in small which introduces small mistakes upon repairing. And if you're targeting the coding DNA sequence, it can basically disrupt the translation of the protein later on. This was also shown. So when you introduce the double-stranded DNA break in presence of a linear DNA, it was also shown that in microalgae, this linear DNA can be incorporated in the target region. So basically, this can also be used for targeted insertion, but still there might be some mistakes on the left and the right end. The another interesting strategy was the homology directed repair. So you basically trigger the double-stranded DNA break, and in the presence of an editing template, this editing template will have a homologous recombination flanks of certain lengths on either end of your desired insert, and this will precisely be introduced into your strain. And these are the basic genome engineering techniques or genome editing techniques that are available for microalgae. So almost 3.5 years back, when I started my PhD, there were only a handful of papers which were using CRISPR-Cas systems in microalgae. And almost all the papers were using the same technique. So they were mostly using a plasmid-based Cas9 system. So basically, they were introducing a linear DNA that was expressing the Cas9 protein and the guide RNA into the host genome. And they were introducing either they were introducing the guide RNA where they want to target. So the guide RNA that is specific to the region they want to target on the same plasmid, or they were doing secondary transformations in which they were putting the guide RNAs inside to generate the mutants. So you always have a continuous or expression of Cas protein, and then you transform the guides. But we were thinking, what if we could use ribonucleoproteins, which have certain advantages? And the ribonucleoproteins are also used in human cells for active gene disruption and also for gene insertions. So the advantages of using ribonucleoproteins instead of a Cas system is that you are transforming a protein as such instead of the plasmid. So the protein is only expressed transiently, because after a few hours the protein is degraded. So it basically reduces the possibilities of low of off targets. And when you are using a plasmid based system, and when you're working on a species like Nanochloropsis, which had less regulatory elements like promoters and terminators, it was difficult to generate a plasmid system that can transiently express the Cas protein and also the expression of the guide RNA. But when we are using a ribonucleoprotein, we don't need this promoters and terminator. We just have to transform the protein. And also at this time back, almost 3 years back in plants, there was a discussion that was going on saying that if you are using proteins instead of plasmid for introducing the mutations based on Cas system, then possibly they might not be rendered as GMOs, which was also an interesting reason to use them. Also, if we can combine the ribonucleoprotein-based approach for with HDR, with homology-directed repair, we should be able to generate precise mutants, which was not the case 3.5 years back. 3.5 years back, almost all the systems were using non-homologous end joining to generate the mutants. So this is the approach that we used. I will take you through the approach that we did one by one. So initially we developed the ribonucleoprotein. So basically the ribonucleoprotein consists of the respective Cas protein and the guide RNA or the CRISPR RNA in case of Cas12a that is capable of introducing the double-stranded DNA break on your gene of interest. As a part of tool development, we always focused on the gene called as nitrate reductase, because once you generate this particular mutant, it was previously shown that this gene is deletable. That was one of the reason, because Nanachloropsis was not what do you say? It was not a model species, or there was no lot of information on molecular tool development on this species at that point. So they did already show that this gene can be deleted and the phenotype can be characterized. So we always targeted the nitrate reductase gene, and we transformed this particular. So when we transformed the ribonucleoprotein, we also transformed with an editing template that had 1 kb upstream and 1 kb downstream homologous recombination flanks. To swap the nitrate reductase gene, the targeting region, with a zeosin resistance, which is an antibiotic that we use for microalgae, to swap the nitrate reductase gene with this particular cassette. So upon transformation, what we expect to happen is that the Cas protein to introduce the double-stranded DNA break, and upon double-stranded DNA break, the host mechanisms will come into play to fix the double-stranded DNA break, and we expect the presence of our editing template to take over at this particular point, and thereby swapping the nitrate reductase gene, which is in yellow here, with the red part, that is the antibiotic-resistant cassette. Once this happens, and when you select the colonies on a zeosin-resistant plate, we should be able to separate the mutant colonies from the wild-type colonies with specific kinds of specific primers that amplify this region. We used various ribonucleoproteins. So we used Cas9 and we used 3 different variants of Cas12a to identify which could be the best Cas protein to be used in microalgae or in specific in Nanachloropsis. And these are the results what we got. So we have different Cas proteins that we tried. And the first part is just the transformation of the homologous recombination template. So when you just transform with the editing template, we were not obtaining any mutants. However, when we combine the editing template along with the ribonucleoprotein, so this is with the most widely used Cas9, we were having efficiencies close to 80%. So the difference between this and this are the spacer sequences. So we were randomly targeting different regions. So when you are using spacer 1 in case of Cas9, we were obtaining an efficiency close to 80%, but with another spacer, the efficiency was around 30%. And also something interesting that we saw was that the AS Cas12a variant was not the best variant to be used in microalgae, because they were really struggling to produce mutants. While FN Cas12a and LB Cas12a, both the Cas12a variants effectively produced HDR-based mutants in microalgae using this approach. However, as I said, when we develop this kind of mutants, we always introduce an antibiotic resistance in the gene, which is not the optimum or which is not the best way of generating mutants. So we were thinking what could be the mechanism, or how could we generate a markerless mutant using the same system. And then we came up with this particular approach or particular strategy. So this is what we previously did. We just transformed the editing template and their ribonucleoprotein for HDR. So what we did was that we, instead of using just the zeosin resistant, we combined the zeosin resistant with the fluorescent protein, so that when you generate the mutants, you should be able to separate the mutants from the wild type using this fluorescence. And we can do a second round of transformation onto these mutants using another ribonucleoprotein that targets the fluorescent protein, and then you use an editing template, but in this case you do not use anything in between, but just the upstream and the downstream homologous recombination flanks. And we proposed that if this happens, there should be at least some mutants or some cells after the transformation that will lose the fluorescent expression, but they will have the color, or let's say in the fluorescence of the wild type. And we should be able to separate them using a FACS system, and then basically grow them on solid media without any selection, because we expect the mutants that we produce by this approach are not going to have any antibiotic resistance. And we can basically, we design the editing templates in such a way that we should be able to differentiate all the mutants and the wild types, and also the mutant that harbors the fluorescent based on colony PCR or the PCR techniques. We basically performed this with two different spacers using FN-Cas12a as the Cas12 as the Cas variant for the ribonucleoprotein, with two different spacers targeting the fluorescence gene. And for one of the spacer, we were, after screening 87 colonies that was sorted out using the FACS system, we did not obtain any mutants. However, with another spacer, we saw that after screening approximately 91 colonies that did not have the fluorescent protein, 4 of them were clear mutants. And as you can see here in the gel image, we were easily these mutants were easily differentiatable on the gel, and we further confirmed the mutants by sequencing as well. So this basically shows that we were successful in developing a ribonucleoprotein-based genome editing systems for microalgae and anachloropsis, and combining HDR. HDR, we showed it for the first time in anachloropsis, and this could generate precise mutants in contrast to non-homologous enjoining based insertion of indels. And also combining this with fluorescent assisted cell sorting can generate markerless mutants in anachloropsis. However, this is not always the best way of doing things. There are always room for improvement, because there were some downsides, because this particular technique always requires purified and active Cas proteins, which can be expensive if you are doing lots of transformations. And transforming with ribonucleoproteins from a laboratory perspective, it's very laborious, because the Cas proteins are always stored in salt buffers, and doing electroporation with a lot of salts in it is not always the ideal way to do it. And for obtaining markerless mutants, we have to do 2 rounds of transformation, taking into consideration approximately 1 to 1.5 months to obtain the colonies after transformation. Will make it quite long. You will need at least 4 months to obtain a markerless mutant with this particular approach, and it is difficult to use this tool for multiplexed genome editing, and also it cannot be used for gene silencing such as CRISPR interference. However, if we develop a plasmid-based Cas12a system, this kind of overcomes most of the downsides of the Cas protein, of the ribonucleoprotein-based assay. Uh, and we decided to go with this approach because by the time we developed the system with ribonucleoproteins, there were already some papers coming out which were describing various promoters and terminators, and also an episomal plasmid system for nanochlorosis. So we made use of all these advancements to develop a Cas12a based or a plasmid based Cas12a system for nanochlorosis. So this is basically the plasmid system that we developed. So this is what you're going to see in the rest of the presentation, where we had a bidirectional promoter that was driving the expression of the guide RNA or the CRISPR RNA with the help of specific ribozyme systems, because ribozyme systems are required for precise generation of the guide RNA, because if there are some extension or if there are some additions into the guide or the CRISPR RNA, it affects the efficiency of the system to generate the mutants. And then you have the Cas12a that was tagged to a luciferase protein, because when you tag your Cas protein to a luciferase protein, at least in microalgae, you can easily detect its expression from a colony level to see whether your protein is being expressed. So to be very sure, we initially used a codon harmonized FN-Cas12a for nanochlorosis. We transformed this plasmid, we selected it on antibiotic, and initially we did an NLEX assay or the nanoluciferase assay to see whether your Cas protein is being expressed. Because if it is being expressed, you should be able to detect the expression of the luciferase protein. However, we were not able to see any expression happening and we did not know why. We were initially confused whether the activity of the Ribby promoter is compromised because we were using this promoter for the first time, and maybe the Cas12a expression is not working enough. We did some experiments to confirm that the Ribby promoter is working well, and then the only confusion remained was regarding the Cas12a gene sequence or the codon harmonized Cas12a variant. So what we did was that we replaced the codon harmonized Cas12a with human codon optimized variants of Cas12a. In this case, we used both FN and also LB Cas12a. The reason why we used a human codon optimized was that previous studies have shown that human optimized gene sequences can be expressed quite well in microalgae. And there were some studies on various expression of various genes in the metabolism of EPA, and also I think for TAC, where they used codon optimized human sequences for expressing genes in microalgae. So we decided to go with these two sequences, and we did the same NLEX assay, and we saw that when we were using these two sequences, we were able to detect the expression of NLEX, showing that when you are using this sequences or this particular optimized sequences, we were able to obtain the expression. And the Cas9 sequence was based, this Cas9 sequence that we used here was obtained from another group, which already did develop a system based on Cas9 for genome editing in nanochlorosis. So this was used as a positive control. And we see that our FN Cas12a is expressed quite well. So we decided to continue our work for generating mutants in nanochlorosis using FN Cas12a that was human codon optimized. In the next round, we transformed nanochlorosis with 3 different plasmids, each harboring different guide RNAs or CRISPR RNAs, targeting random 3 sites in the nitrate reductase gene. And we removed the luciferase or the NLEX protein initially, just to make sure that it is not interfering with the activity of the Cas protein or the activity of Cas protein to introduce a double-stranded DNA break. And we obtained quite interesting percentage of mutants from approximately 90 to 96 colonies that we screened. And we obtained up to 40% of mutants with this particular approach. Then what we did was that we wanted to see whether the introduction of the NLEX back into the Cas protein will have an effect, because we can isolate or we can easily screen the colonies that have the NLEX expression. So what we did was the same. We added the NLEX back into the Cas protein. We transformed and we added the additional NLEX assay step, where you screen only the colonies that have good expression of NLEX. And then we checked whether we were able to obtain the mutants. And this basically, with this particular approach, we enhanced the efficiency in which we obtain the mutants by up to 90%, which is quite interesting and which is quite efficient from a genome editing perspective. Next, uh, I hope you are still able to hear me, because I'm getting a message here that my internet connection is unstable. Are you still able to hear me?
Yes, we are following you very well.
Hello?
We
Okay,
can
perfect.
follow you very well.
So as I said, okay, thank you. Yeah. So as I was saying, we were using hammerhead, and we were using ribosome systems for precise production of the CRISPR RNA. However, this was hampering the plasmid development and also the possibilities of performing multiplexing in microalgae, because these hammerhead ribosomes require specific sequences at its 3' end and also at 5' end for precise generation of this particular CRISPR RNA. So if we can get rid of this particular ribosome systems, that can again elaborate the possibilities of using Cas12a for multiplexing and all the other advantages that Cas12a have. The hammerhead ribosome from the 5' end of the CRISPR RNA. In this case, we only used one of the spacer that worked the best in our previous experiment to see whether the system is working. And with this, we were able to further improve the efficiency to 95%. So even removing the ribosomes generated mutants with very high efficiency, showing that the ribosome systems are not required when you are using a Cas12a protein. However, just to keep in mind, for Cas9, it is quite relevant because Cas9 doesn't have the ability to process its own pre-CRISPR RNA, but Cas12a has, which can be further exploited for multiplex genome editing. So multiplex genome editing means you target multiple sites in a single transformation so that you can generate multiple mutants in a single transformation. So for this, we just to have a proof of concept, we targeted two different sites in the nitrate reductase gene. So basically, if they targeted two different sites, possibly the region in between might be deleted, or they might introduce singles at both the sites. We used the two spaces that worked really well in our previous experiment, where we used single targeting. And upon transformation, we saw that 35% of the mutants that we screened had mutations at both the sites, and another 30% of the mutants harbored mutation at one of the target sites, showing that the multiplexing also works quite well with this particular system. And we were showing this for the first time in any of the microalgae using, not we were showing for the first time that CRISPR array can be efficiently used for Cas12a based multiplexing in any of the microalgae. Next, we wanted to further see whether we can target more regions. So we introduced another spacer sequence in there that was targeting the LPAT gene, which was involved in the production of triglyceride. However, we did not have any phenotypic characterization of this particular mutant, but still it was shown in literature that this gene can be knocked out. So what we saw was that we were able to obtain mutants with an efficiency of 14% for all the three targets. And we had an efficiency of 4% of at least any of the two targets, and 13% of the mutants had mutations at any one of the target. This basically shows that you can further extend the CRISPR array into how much or into how many lengths you want, and based on how many targets you want to target in a single transformation, and the efficient self-processing capability of CRISPR RNA by Cas12a can get the job done. So to summarize this part, we developed an efficient FN-Cas12a based genome editing system in Anachloropsis using ribonucleoproteins, and also based on Cas12a based on plasmid. And we show that when you are using Cas12a, you can get rid of, or you don't really have to use the ribozymes. That really simplifies the plasmid development, and also efficient multiplexing is also possible with this particular system. Next, we wanted to develop a tool for gene downregulation. So the Cas protein, it can introduce double-stranded DNA breaks. It has, as John mentioned, for Cas9, it has two catalytic active sites, and for Cas12a, it has just one. If you introduce specific mutations in this particular system, it will bind to the target DNA, but it will not introduce double-stranded DNA break. And it was shown that if you introduce these mutations in the live Cas proteins, it binds to the DNA and it can block the transcription machinery. If you target the promoter of the gene, it blocks the transcription machinery from initiating the transcription, thereby downregulating the gene expression. And if you target inside the gene, still it blocks the transcription, and still it reduces the transcription of the targeted gene by downregulating its expression. For Cas9, these are the mutations that we introduced to obtain a dead Cas protein. We call it dead Cas because it cannot introduce double-stranded DNA break. And for developing a dead Cas12a, these were the two mutations that we did. So our approach here was pretty straightforward. We had the dead Cas protein that we transformed into the fluorescence expressing strain that we developed for our first study. And you basically quantify the expression of TD tomato, or here TD tomato is the fluorescence that we're talking about. And based on the differences in the fluorescence level in the controls and also in the targeting, you should be able to see whether there is an effectivity happening in the downregulation. So We developed tools for both Cas12a and also for Cas9. We developed at Cas9 and at Cas12a with multiple spacers that were targeting multiple regions in the TD tomato gene. So we transformed it into the fluorescence expressing strain. We selected an antibiotic. We initially performed NLEX assay to see or to take specific colonies that have good expression of Cas12a and Cas9. We scaled up the cultures and measured for fluorescence and other things. So these are the fluorescent results that we have. So when we were using Cas9 alone and also Cas12a alone, this is the negative control on the graph. This is the normalized fluorescence that we get. And these are the different regions where different spaces or different CRISPR RNA or sgRNA targets. As you can see here, compared to the negative control, we don't see an efficient silencing because the normalized fluorescence are higher or it was similar to the negative control, showing that when you use that Cas9 and that Cas12a alone, we were not able to obtain efficient silencing in Anaphylaxis. From literature, we found that at least in certain certain eukaryotes, using Cas9 or Cas12a alone for CRISPR interference might not be the best approach. However, you will have to tag them with um certain transcription repressor domains to achieve proper silencing. So we decided to use CRAB, which is one of the most widely used transcription repressor domain for CRISPR-I. We attached it to the dead Cas proteins and repeated the same experiment. So here now this is the complex that we have. We have the Cas protein tagged with the transcription repressor domain or CRAB, and again with the NLEX. Now you have a big chunk of machinery going in there. And we followed the same approach. And now we started seeing some repression, because you can see that compared to the negative control, we were able to obtain like 40% to 20% repression from the fluorescence level with Cas9, and we also saw up to 50% silencing when we were using the Cas12a based on where we were targeting. Something that we saw, something that we found interesting was that when you target very beginning of the gene, so here we can see that sgRNA2 and CRISPR-RNA2 targets in the beginning of the coding DNA sequence of TD tomato, and that is where we obtain or where we observe the uh, best down regulation in the fluorescence of TD tomato. To further confirm that the results that we see, to further confirm this from a genotypic perspective, we isolated the RNA, we performed the cDNA synthesis and performed qPCR to confirm that this effect is reflected from an mRNA level. And this is what we observed once we did this. So for the dead Cas9 along with CRAB, we were able to obtain up to 90% repression in the TD tomato expression or in the TD tomato mRNA levels when you were targeting in the very beginning of the gene, and up to 60% silencing was obtained when you were targeting in the beginning of the gene with Cas12a as well, showing that CRISPR interference works well when you are attaching the Cas proteins with the CRAB, and it works the best, at least for us. In our case, it was working the best when you are targeting inside the gene in the very beginning of the coding DNA sequence. So to summarize, we developed an effective CRISPR interference system for microalgae and also for Nanochloropsis. And transcription repressor domains are quite important to achieve this. Without it, it might not work. And CRAB might be a promising candidate for this. There are other transcription repressor domains available, but we haven't checked it. But there might be some other variants that can work as well. And considering the positional effect that we saw, maximum silencing, what we observed was in the very beginning of the gene, but multiple spaces or multiple targets can be tested to see, because in the literature you see that targeting the promoter might be the best, but you also see the other way around. So I think you will have to target different, you will have to use much more spaces if you really want to see where the best silencing is achieved. So to finally conclude, we developed a markerless and precise genome editing system for using Cas ribonucleoproteins and homology directed repair for generating precise mutants. We also developed a plasmid-based Cas12a system for Nanocleropsis oceanica, which is much more efficient than using Cas9 systems. We have also reported multiplexed genome editing using CRISPR array, targeting up to 3 different regions in a single transformation for the first time in microalgae, in any microalgae, not only Nanocleropsis. And also we developed an efficient gene silencing tool for Nanocleropsis, which can also be adapted to other microalgal strains. So I would like to thank my supervisors and collaborators. And also my project is funded by a Magnificent project from EU. And if you have any questions, I will try to answer them. Thank you.
Thank you very much, Mikris. A very impressive work. I mean, did you done all this in just 3 years?
Thank you.
It's really impressive, but
Yeah.
having such an advisor as Professor Bangar
I mean, we had a lot of help. It was not me alone. Like we had really good students working along with us.
Yeah. So I would like to kindly ask.
So yeah, that really helps at times.
Professor Kifiriuk, if you have some questions, because I know that you have to leave very soon.
Yes, unfortunately, I have to leave the meeting at 1 o'clock. This was a very nice presentation. I think that it is very valuable as a model for our students attending this presentation. What I would like to ask Miris is if you made an assessment of the financial fee support needed for developing all this experimental setup, and which is the average time needed from the beginning, from setting up, I don't know, an idea until obtaining a specific CRISPR system dedicated to a certain application.
Okay. So I think you are asking me how long will it take since we start developing. So
Yes.
once we start from scratch, how long will it take until we develop a complete genome editing system, right?
Yes, yes.
So the toolbox
Yeah, okay.
was developed
I would say yes.
by your group, yeah.
Yes,
Yeah.
considering
I would
that
say this depends. Yes, you may go, please, Professor.
no, it's okay. I was saying, considering that we are talking about an in vitro application, not an in vivo application.
Oh, I mean, I'm not really sure. What do you mean by in vitro applications?
For example, not involving an animal or human individual to receive
Oh,
this.
okay. I would say how long will it take is if you are for example, in case of in vitro, and if you want to make some changes, I think it shouldn't take long because everything is happening inside a test tube, hopefully. But if you are talking about in vivo for developing an entire genome editing toolbox for a new species, I would say it depends on a lot of things. First thing is the doubling time of the species, because in my case, for me, after transformation, it takes like 1 to a bit more than 1 month to obtain the colonies and to see whether my system is working or not. So it took some time for me to develop the tools as such. But let's say that you are working on something like E. coli, where you do a transformation and you get the colonies the next day. It will take much less time. For me, it took approximately 1 year to get some positive results and see that systems are working, and another 1 year or so to develop all the multiplexing and the CRISPR interference and all these things. But if you're working on a fast growing species, I think in 6 months, I mean, if you are being really optimistic, I mean, 6 months to 1 year should be sufficient if you already have a proper Cas9 expressing sequence, and if you have good regulatory elements like promoters and terminators to control its expression.
Thank you so much.
Yeah.
we thank you, Miris. So we have also the question and answer, let's say, box for those who would like to interact and to ask questions for this magnificent work done by the team there at Wageningen. So from my perspective, I was wondering when you have focused on a specific type of microalgae, what kind of motivation you had for that?
So Nanachloropsis oceanica is considered to be an oleaginous microalgae. So basically, this particular strain can accumulate large amount of lipids, triglycerides, I would say, which is used for biofuel production. And there are already quite some research going on in this particular strain to see whether we can commercialize this. And one of the main bottleneck that we come upon is that the present amount or the present productivity of lipids are quite low, that they cannot compete, or the present production cost is too high to meet the present market demands or to compete with the fossil fuels. So the hypothesis is that if we can somehow improve the production of or the productivity of these lipids, we might have a sustainable source for producing biofuels from the species. And there are quite some research happening on the species, not only in our lab but also in some labs in US, in Germany, in Australia. People are trying to see whether we can improve this strain to make it produce more of lipids from it. Not only lipids, presently this strain is also used for producing high value compounds which are commercially feasible. So they are producing omega-3 fatty acids from this particular strain, and it is already in the market from what I can understand, as nutritional supplements and things like that. But the present research is also, there are also research groups that they are focusing on improving the production of this omega-3 fatty acids, and always having a good or high throughput genome editing tool will always accelerate the process of developing mutants and how quickly you can develop new strains. So that was the idea of using this strain as the strain of interest for our study.
In our case, following the support of Professor van der Oost, we had a discussion with Professor Barbosa, and we were evaluating, because we have a project on biofilms. You know, at the wastewater treatment plants, you have this process of aeration, and alternatively for let's say the depletion of different components in the water, there is technology based on biofilms. So these are some plastic bodies on which there are different compounds of bacteria, and these bacteria are established based on the substrate that is there present in the pool. So the idea was if it is possible to identify different strains into this mixture of bacteria, and then try to improve this mixture in order to increase the efficiency of working on this, let's say, treatment process. And of course, the whole problem is how to identify a specific targeted bacteria or a specific targeted microalgae, and to know also what is the potential of improvement of that specific microalgae or bacteria in the direction that we discussed. For instance, if you want to evaluate the possibility that a specific strain could contribute to the decomposition of some sort of, let's call it, biostimulators or different types of pharmaceutics that are present in water, or things like that that could be targeted. But it is very difficult to identify first, to see what kind of sequences are, let's say, potentially operational for something like that. And then, of course, as also Professor van der Oost mentioned, everything is possible, but first of all, you have to know what you want, and
Yeah.
second, what kind of price are you ready to pay for that?
Yeah. I mean, I would say if something like if we are really looking forward to isolate a particular algal strain or a bacterial strain that can decompose a particular component, I would say, I mean, this is what John would also say. Like, we will have to look into the nature and see where these components are present and try to isolate strains from that environment. As you are saying, some kind of biostimulants. I mean, if they are dumped somewhere, I'm pretty sure that there might be some bacteria growing in in that environment, and we will have to isolate it and see what bacteria it is and what it is doing. And yeah, and then maybe study what is happening, and if we can tweak it somehow to use these particular compounds more, I think that would be the way to go.
Yeah, because for instance, you know that the problem of hormones, of pharmaceutics, of antibiotics present in the wastewater is an issue that at present has not a solution yet. So from that perspective, detecting them, understanding what can be done, and then developing these techniques for putting bacteria and microalgae to work, it is a potential area of development.
Yep, definitely, yes.
So regarding the upscaling of these technologies, because of course you developed these, you made a proof of concept. So now what kind of valley of death you have to further pass in order to have them as a proved technology for application?
Oh, I mean, I would say the next step in this particular research would be to further look into the metabolism. So in my perspective, from the perspective of looking into nanochloropsis, I would see we'll have to look much more into detail into the metabolism of nanochloropsis and see what is resulting in producing the lipids. And we have now efficient genome editing tools that can introduce multiple mutations in a single round of transformation. We have efficient tools that can downregulate the expression of certain genes. So I would say we will have to make use of what we have developed quite well and try to develop strains that can efficiently um, channel the carbon that it is captured towards lipids and triglycerides. But from what we understand, that might not be the best way to go, because what we understand is that the carbon dioxide, that is one carbon that is coming inside, is one of the bottleneck, or it is not the best way to use the atmospheric carbon, or we have to find ways to improve the carbon capture and also improve ways to make use of the absorbed sunlight energy in the best way possible and channel it towards improving the entire productivity. And just focusing on one product by making some small changes in the downstream part of the metabolism might not be the best way to obtain a mutant or obtain a strain that can address the big challenge of producing biofuels from microalgae. And there can be a lot of questions. You can always ask whether nanochlorosis is the best strain towards it. You never know, because I mean, last year or maybe 2 years back, they even found a microalgae that can double in 2 hours, which is very interesting when you look from the perspective of a phototrophic organism like an eukaryotic phototrophic organism. Because in our case, it's a bit more than 20 hours, and then you have something that grows 10 times faster. Then you think whether you should put the entire pathway of producing lipids into a fast growing strain, or you should look into the fast growing strain and bring something from that strain into a slow growing strain. So I think there are a lot of questions still to be answered, and I think developing these tools will help us answer these questions quite faster.
Thank you, Nedis. Waiting for other questions, I would like to come back to Professor van der Oost, and I would like to ask him from, let's say, fundamental research point of view, the further development of CRISPR-Cas. What kind of new path you would suggest for the youngsters that are present here and would like to take the future Nobel Prize?
Well, I wouldn't think about future Nobel Prize. I think for my advice for the new generation would be to follow your heart and just go for the kind of research that you really like a lot, and then go for it, and try to be creative, think out of the box, and don't think about prices. Just think about trying to discover new biology, and so that's what we did, and we were lucky enough to stumble upon this whole miracle of CRISPR-Cas, a beautiful outcome of the evolution, or the microbial arms race. And I think, well, what I said, I mean, new subtypes of CRISPR-Cas are still being discovered. Of course, you can imagine that at some stage we will reach a plateau. So I don't know how much further this will be going on. But apart from that, a couple of years ago, there was the group of Rotem Sorek from Israel, from the Weizmann Institute, and he kind of focused on the so-called the clusters. How did he call it? It's not pathogenicity islands, but at least it's clusters of genes where there are a lot of defense systems. So including the restriction modification systems, including CRISPR, but also a lot of unknown genes are still out there. And that's what he looked at, and he kind of transplanted some of these clusters to bacillus, for instance, or to E. coli, and then found that they gave rise to immunity as well, meaning that what we now know is just the tip of the iceberg. And I'm not saying that all these systems will be as exciting as CRISPR, or well, they may be as exciting, but maybe not so easy to translate to all kind of applications, because of course that's the beauty of the CRISPR system. I've said it already a couple of times, that the fact that you can easily adjust it just by changing the DNA coding for the CRISPR guides. That's so easy. You can do that in a couple of days, and then you have a new nuclease with a new specificity, right? And that is not always the case with the other defense systems. So like the restriction modification system, there the specificity relies on the protein. So then you have to do protein engineering to adjust the specificity, which is with the current tools and the current insights is a project on its own, right? I mean, but also if you look at the CRISPR literature, for instance, I mean, spectacular recent developments, I would say, are the engineering of synthetic CRISPR variants, like the base editing and the prime editing systems developed by the group of David Liu at MIT in Boston. That's beautiful, but there may be more variants that you can think of, right? So just exploiting the specificity of these systems, and then you can basically fuse any functionality, any enzyme, any protein that you like to that CRISPR nuclease, and just use that for whatever, for detection purposes or for editing purposes maybe. So just be creative. People have said that the only limitation of the CRISPR-Cas system is our own imagination, and so I have faith that many people that are in the audience now are bright people, have wild ideas. Just go for it and try, and maybe you're lucky, and so I wish everybody all the best of luck in their future careers.
Thank you, John. Another question that I had in mind from the beginning of your presentation, because how can we consider the influence of such a technique, a technology, being present in nature, and how much of the let's say evolution in nature and diversification of life in nature was how can I say, or could be explained for some mechanisms similar to this one?
Yeah, well, that's a good question. Well, again, it's all evolution. Everything we see is the result of evolution, and especially and also the diversity we see in the different CRISPR-Cas systems. They all reflect basically the ongoing warfare between phage on the one, or mobile genetic elements, including viruses and phages on the one hand, and also the bacteria and the archaea on the other hand. But there is more to it. Also, I mean, we recently published a paper with colleagues from Rotterdam in the Netherlands, where we showed that Campylobacter, so a pathogenic bacterium, that also uses its Cas9 to really, after invading the human epithelial cells, that the Cas9 can, without any synthetic tags or so, can find its way to the nucleus and there detect or destroy DNA, right? So there is much more to this. So also, I mean, a couple of years ago, anti-CRISPR systems have been found. So small proteins from phages that can kind of disrupt the functionality of the antivirus system. And then as a next step in the arms race, also recently anti-CRISPRs have been found. So trying to counteract these anti-CRISPR systems of phages. And so and that tells you that there is really a huge selective pressure on trying to come up with new variants, and that's exactly what you see if we now look at the overview of the different classes, different types, different subtypes. It's overwhelming, but that's just the result of the ongoing arms race. So and that will not stop, never. Like the new viruses that threat us. That will never stop. But with all our insights now, and that will continue, and that for that the upcoming generations are going to be very important. That we are better and better capable of reacting to things like that, just as it happens in nature. And of course, we use our brains and our we stand on the shoulders of the people that made very important discoveries to climb up and to be able to react faster and faster and more efficient to all kind of threats. But it will never stop, so we have to keep on going.
To make it more complicated, I would like to tell you about a friend of mine, Professor Laughlin from Stanford University. He also won a Nobel Prize for Physics. And he's working on the interaction between electric and magnetic fields on the cells, including the aspect which is actually annoying Miris in the time of the application. So in order to decrease or modify the time of the application of different cells, he wanted to submit the cells under different type of fields, electric fields, magnetic fields, and things like that. So from that perspective, do you see a possibility for the future to combine these two aspects? I mean, to take a sort of interaction at the level of different, let's call them chemical at the end, sequencing of the DNA or RNA, and to combine them with an applied, let's say, combined electric or magnetic field at that level.
Sure. Well, as said, I mean, many things are possible, and I think we all should have an open mind about maybe at first sight kind of weird combinations, but sure, I think so. So on the one hand, the cells rely on their genetic information, but of course, I mean, it also depends, or the fitness of a cell with its genome and with all the cargo of genes. The fitness depends on the conditions, right? And if we change the conditions by maybe using electric field or maybe even a magnetic field, that could all influence the phenotype. So how the biological system reacts to that. So indeed, I could imagine that if you get the right tuning of the genotype with those conditions, that you may get very interesting phenotypes, but it will be very difficult to predict how we should adjust our genotype. So either go and what Miris also indicated, go and find some interesting genetic variants in nature, or do random mutagenesis to adjust them and luckily at some stage find a right combination that does a certain trick, right? But it will be very difficult to really, or at least my brain certainly is not capable for doing that, to come up with certain adjustments at the genome level to make a certain bacterium do a better job under a certain electric or let alone magnetic field. So for that, I think again you should use the principles of evolution. So you or generate random mutants, or go and look in nature and then see what variants perform as you like them to perform under certain conditions. So indeed, I think it's good to have an open mind and think along those lines, but it won't be easy. But well,
yeah, because there were facts. I mean, when Bob was telling me about his works, that, for instance, a question that could be out of the mind of non-biologists or non-genetics specialists is how a cell is finding exactly the middle for duplication, for instance. So why is so symmetric and so on. So under different type of fields, then there were present the asymmetries in the replication process and things like that. So it is clear that there is an influence and should be an influence. And exactly as you pointed out in the slide that has been shown by Miris, these aspects might go into this also, let's say, oriented type of intrusion into genetics, or further out in facilitating the evolution of a specific type of mutant to go further. So that's clear. But on the other side, you know, there is, because your work mostly is in the area of sci-fi, so usually you put yourself this question, this pattern and how can I say, haplotype of life that exists on Earth, and we are here, is actually at the end the result of evolution of different type of sequences of DNA and RNA coming from somewhere in somehow. Would the gravitation, the magnetic field, the electromagnetic fields play a role in shaping the life here? So this kind of life that we are living and we are part of is not an influence of all these evolution process but evolution that has been, in some sense, a result of influences of this kind of, because it's what we know, the fields or the matter.
No, that's a very good point. And another to refer to another Nobel Prize winner, that's Frances Arnold. So she got the prize for developing laboratory evolution, right? And she one of her quotes is you get what you screen for. So depending on the screening conditions, you may end up with certain variants, certain genotypes that perform better. But if you change the conditions, then you get something else, right? So indeed, on another planet far away from here, there will be different gravitation, different chemistry, maybe different solutions to store genetic material, and that led to I guess many different outcomes, but I'm afraid that during my lifetime I won't be in a lucky position to see how that looks. But that would be great.
Yeah, but at least I'm sure that some of the students that have followed this course, they will have this chance to face it, and
I hope so. I hope so. Yeah.
And they will remember about your course and the presentation also of Miris, and about the major, let's say, takeout from here that you mentioned, to be open and to fight for what they are thinking, and then they will succeed to do something in that. But besides of that, of course, this planet is in a peril, and we all know about this unbelievable type of pollutants and unbelievable type of degradation of the habitat and so on. So we have to find, and we need to find some solution in order to live in, because otherwise very soon, maybe even in our lifetime, we have to look for another planet.
I don't think so. Of course, I mean, I completely agree with you of all the serious threats that are often the result of human activity. But I think we also have the brains that we should do something about that. So let's hope that also the people that are attending this meeting, that they also in the future, that they work all together on trying to make this a better place, or at least to make sure that it will be a planet that also future generations can live on, and that they have clean air and clean water and enough food and things like that. And to some of these things, I think at least on the short term, CRISPR could definitely contribute. And we started talking about this before the meeting started. I mean, if you now see that kind of the applications of CRISPR in the plant biotechnology field, that they are really being obstructed by the policymakers, because basically they don't know what they are talking about, but that really could obstruct the use of CRISPR to provide sustainable food for everybody on this planet, also in the future. And then I think indeed, as scientists, we should take our responsibility to explain not only to the general public, but also to the policymakers that we shouldn't waste a lot of time. And when it comes to a pandemic like we are facing now, then you see that they are ready to move faster than they are used to, and that all the rules and regulations then that is put aside to develop these vaccines that we are now using. But also this moment is getting closer and closer, when it comes to global warming. It comes closer and closer when it comes to food production for future generations. So we really, as scientists, also the people in the audience, we should all together, we should take our responsibility and well, reach out and make this happen. And
yeah, and besides of that, what the history has shown is that when you put some artificial barriers like legal, whatever kind of things in the front of the eager for knowledge, for scientific research, for the curiosity of going beyond. So this is not blocking something. It will go further for sure in some other places in the generating different aspects that will be not necessarily pleasant for you. For instance, it was the STEM technology, you remember years before, then it was also threatened in different places, but then it erupted in China or erupted in Ukraine, in places where there were no regulations and things like that. And then this technology went further in those places, and then Europe was in the need to reconsider everything, but to take it from a very low step forward. So from that perspective, there are different aspects that have to be, let's say, reconsidered in some way. Of course, it should be always a balance. You know that here in this part of the world, the Greeks, they have said that the golden way is the middle way.
Sure.
So you always have to find that middle between, of course, considering the concerns and not leaving that without any kind of control, and on the other side to open and keep the possibility to go further in research activities, to go further into the creativity, and in finding out what kind of really positive aspects can be used, as you mentioned, for the major problems, major challenges of the humanity.
Mm-hmm.
Okay, so once again, thank you for your time, for your efforts. It was quite a challenge also to make this course from your side, and we would like to thank for your generosity to accept to be with us and to share your knowledge and your vision. We hope that this will be just a beginning, because we hope that also we shall send you different questions, we shall ask you for different possibilities, and if it will be any chance, we hope that we can develop some projects together.
Mm-hmm. Okay. Well, it was a pleasure to participate together with Miris, and we will be. Well, it was fun to do. I hope it inspired many people, and indeed we should look at possibilities in the future to collaborate or whatever.
Thank you very much. Thank you for everything.
Bye bye.
Bye.